Biological molecules are the substances that make up living things, whether it's humans or bacteria or plants or animals or fungi. And there are four types of biological molecules. Today, we're going to be talking about one type known as lipids. And by the way, if you find this video to be of help, please subscribe to my channel, and that will help other people find this video as well. So within the class of lipids, we're going to be talking about neutral lipids. Neutral lipids, specifically fats and oils. And fats and oils can be made up of what's called saturated fatty acids, or they can be made up of unsaturated fatty acids as well. We're going to talk about that and break down what all this stuff means. But remember, neutral lipids like fats and oils are non-polar, okay? That means they're hydrophobic. Water is polar, so it doesn't associate or mix with things that are nonpolar. And so because lipids are nonpolar, that makes them hydrophobic, right? They don't associate with water. And that's why maybe if you have a salad dressing and you let it sit for a while, you'll see that the oil starts to separate, right, from the water or the aqueous part. The oil is less dense, so it floats on top. But the reason that they don't mix together is because oil is hydrophobic, because it's nonpolar. So this is a summary of what I'm going to break down today. Okay, I'm going to break down that fats and oils are neutral lipids. Fats and oils have a structure which is known as a triglyceride. Triglycerides are made up of a glycerol molecule that's bonded to three fatty acids, and fatty acids can be saturated or unsaturated, depending upon whether they have any double bonds in their hydrocarbon portion. So I'm going to break this all down for you today. So this is what's called a space-filling model, and it is a model of a lipid, a neutral lipid like a fat or an oil. Okay, and in the space-filling model, oxygens are in red, carbons are in gray or black, and the hydrogens are in white. So you can see that in oils and fats and these neutral lipids, there are only a few oxygens. Most of the molecules is composed of carbons and hydrogens. And this particular structure is known as a triglyceride, which is also known as a triacylglycerol. This structure here, this triglyceride structure, is the main form in which oils and fats exist in living things or in food, right? Because the things that we eat, we're living things as well. Now, it's called a triglyceride because of the different parts of this molecule. The part at the top that has some oxygens on it, that's the part that is known as glycerol. And I'll show you a better structure of that in a minute. The three parts that hang down off of that glycerol portion are known as fatty acids. Okay, so in every triglyceride, there is a glycerol part. That's the glyceride part of the word. And there are three fatty acids attached to that glycerol. And that's the tri part of the triglyceride. What happens is that the three fatty acids become bonded to that glycerol backbone, and that's when we call it a triglyceride. So here we're looking at a little bit more of the molecular structure of the molecule. And we can see here over on the left that this is the glycerol molecule, right, which is a carbon backbone. What does that mean? That just means it provides the structure for the molecule that's going to be made, the triglyceride. Glycerol has three carbons. Now, over on the right, we see three fatty acids, okay? 1, 2, 3. And the glycerol and the three fatty acids are going to bind together. And it's those atoms that are in blue that are going to actually be removed in the process. Now, you might notice those atoms in blue, we have an H in the glycerol side and an OH for each fatty acid chain. Those are going to be removed. And what is that? That's H2O. Okay, so when glycerol bonds to these three fatty acid chains, it's actually going to release three molecules of water. And because it releases water, we call this a dehydration reaction. And we can see that that's happened here. So over on the right side, we see there's that glycerol molecule now bonded to the three fatty acid chains. Now we call it a triglyceride molecule. And if you're interested, each link is called an ester linkage. Okay, so if you take an organic chemistry, or maybe are familiar with some of these words, an ester linkage is when a hydroxyl group binds to a carboxyl group, and that's what we have happening here when that fatty acid binds to the glycerol molecule. So here we have the triglyceride, that glycerol bonded to 3 fatty acids. This is another way of looking at the molecule. We're looking at a line drawing here. And here we can see that was the glycerol part of the molecule, right? With the 3 carbons and the 3 oxygens. And then we have the 3 fatty acid chains that are attached to that glycerol molecule. Now, in this case, all of the fatty acid chains are exactly identical. They all have the same number of carbons, for instance, but that's not necessarily the case in living things. This is just an example. So different organisms and different types of fats and oils will have different types of fatty acid chains attached to that glycerol backbone. So here are some models of the different fatty acid chains that we can find. Now we're just looking at the fatty acids, right? And you can see on the left side of each of these molecules, you can see the 2 oxygen molecules in red, and that helps to orient us that we're looking at the part that binds the glycerol molecule. Okay, so when you see those 2 red circles, those are the oxygens. That's the part that's going to interact with the glycerol molecule. But you can see, looking at these different fatty acids, that they're not all the same. There are lots of different types of fatty acids that have different numbers of carbons in them, that have different shapes and structures, and that have different sizes as well. So depending upon the particular fat or the particular oil, there's going to be specific fatty acids that are found in those 3 positions. And it doesn't have to be all the same fatty acid. It could be different fatty acids in these fats or oils. So now I want to talk about those fatty acids specifically, right? We have 3 fatty acids that bind to the glycerol molecule. Now, fatty acids can be known as saturated, or they can be known as unsaturated. And if they're unsaturated, they can be known as monounsaturated, or they can be polyunsaturated. So let's break down what that means and the structure of fatty acids. Okay, so here is a fatty acid. I flipped it around so you notice that those oxygens are in red are on the right side. Okay, that's the part that binds to the glycerol molecule, but we're looking at a fatty acid. This happens to be myristic acid, which is a fatty acid. Now let's look at the structure of a fatty acid. Okay, this is a line drawing of the structure of a fatty acid. And I've made it a little bit smaller so that we can actually fit it here on the screen. But within each fatty acid, there's the part that has the 2 oxygens, right? There's a carbon that's double bonded to an oxygen, and then that carbon that shares a single bond with an oxygen, which bonds to hydrogen, right? That particular structure is known as a carboxyl group. If we're looking at the space-filling molecule, the carboxyl group is the part that has the 2 red oxygens in it. Now, the rest of the molecule is all carbons bound to hydrogen, and that's called a hydrocarbon. A hydrocarbon is simply a molecule made up of carbons and hydrogens. So this part of the fatty acid is a hydrocarbon. And this is the part that actually will change, okay? So the carboxyl group will remain the same from one fatty acid to another, but the hydrocarbon can change and can get longer or shorter or change its structure. Now, what we're looking at here is known as a saturated fatty acid. Saturated fatty acids have the maximum number of hydrogens bonded to those carbons within the hydrocarbon structure, okay? So the hydrocarbon part of the fatty acid can change, and here we see that every carbon in the hydrocarbon portion is bound to 1 or 2 hydrogens, okay? There are single bonds that are bonding the hydrogens to the carbon. Now, I want to give you a reminder about carbon and single versus double bonds, okay? As a reminder, carbon has 6 protons, 6 neutrons, and 6 electrons. Now, those 6 electrons are going to circulate around the atomic nucleus that contains the protons and the neutrons. In the first shell that's closest to the protons and neutrons, there can be 2 electrons circulating. Now, in the second shell, well, that means that the other 4 electrons of carbon are going to be in that second shell for a total of 6 electrons. Now, in that second shell, you can have a maximum of 8, okay? So carbon has 4. We call the electrons in the outermost shell the valence electrons. So carbon has 4 valence electrons. But atoms are always trying to interact such that they're in the lowest energy state. And that means that they have a full complement of electrons in that outer shell. Now, carbon has 4. So 8 would be the maximum. It will participate in bonds until it reaches the maximum of 8. Now, carbon can participate in single bonds, double bonds, or triple bonds. A single bond is when there's going to be a covalent bond between carbon and another atom. Now, covalent bond means that they're each going to share one of their electrons. So in this particular example, we see this carbon. Now, this carbon already has 4 of its own electrons. It's going to share one with hydrogen. It's going to share one with this hydrogen. It's going to share another with this hydrogen, and it shares another one with this carbon. So carbon here starts out with 4 of its own, and it shares 1, 2, 3, 4. Okay, so now it has a maximum of 8 in its outermost shell. Now it's called a single bond when each atom shares 1 of its electrons with the adjacent atom. Okay, so all of the bonds shown here are single bonds, and we note that by doing a single line in between the two atoms. Now, carbon can also participate in double bonds between the carbons. So here's an example of a double bond. Now, again, carbon has 4 of its own electrons, and it participates in a covalent bond. So it shares one of hydrogen's electrons. It shares one of this hydrogen's electrons. And here it shares 2 electrons with this other carbon. Because each of these are sharing 2 of their electrons, we call that a double bond. And as you can see, we draw a double line to indicate that they're sharing 2 of their electrons. So this carbon starts out with 4 of its own. It shares 5, 6, 7, 8, right? It shares 4 with the atoms around it for a total of 8 electrons now in its outermost shell. And that forms a very strong bond. So a single bond is when carbon or other atoms share 1 electron with an adjacent atom. A double bond is when 2 electrons are shared. And we see here a double bond between these 2 carbons. They're each sharing 2 of their electrons with the other atom. Now, I want you to notice one other thing. When there's a double bond, such as in the right example, because that carbon is sharing 2 electrons from the other carbon, it doesn't have any room for an additional hydrogen to bind. So in the left example, we see the carbon is sharing a single bond with this carbon, and then it's bound to 3 hydrogens. Because in this right example, the carbon shares 2 electrons with this other carbon, well, now it actually only needs to have bonds with 2 hydrogens. Okay, so whenever a double bond appears, that's going to reduce the total number of hydrogens because that carbon is filling up its outermost shell, sharing electrons with another carbon through a double bond. And so there are fewer hydrogens that are able to bind to that carbon. Now, bringing it back to our example of fatty acids, we see this structure, right? This is known as a saturated fatty acid because the hydrocarbon part has the maximum number of hydrogens bonded because those carbons are participating only in single bonds. Now, again, disregard the carboxyl part because that's going to be the same in all of the different fatty acids. We're looking specifically at the bonds between the carbons and hydrogens in the hydrocarbon part. And a saturated fatty acid is only going to have single bonds between those carbons and hydrogens. Okay, now I want you to look at this structure, this saturated fatty acid, and see if you can notice the difference between this structure and this structure. Okay, structure 1, structure 2. Structure 1, structure 2. And yes, I'm sure you've noticed, look, the hydrogens there have disappeared, and that's because these 2 carbons now share a double bond. Okay, so because the carbons are sharing an extra electron each, there's no room for extra hydrogens. And so this is known as an unsaturated fatty acid. Okay, when we look at a fatty acid that in the hydrocarbon portion has a double bond, that means that it's unsaturated. It could have more hydrogens if that were a single bond, but because it's a double bond, there are fewer than the maximum number of hydrogens in this hydrocarbon part, and so we call it unsaturated. Okay, so saturated fatty acids are only going to have single bonds between the carbons in the hydrocarbon part of the fatty acid. Unsaturated fatty acids are going to have double bonds. And that's it. That's the difference, really. So I know these words sound really complicated and technical, but the difference between a saturated or an unsaturated fatty acid is whether there are any double bonds in the hydrocarbon part of the fatty acid. Now, we call this particular fatty acid a monounsaturated fatty acid. Monounsaturated fatty acids have one double bond, okay? So unsaturated fatty acids, it means that they're going to have a double bond in that hydrocarbon part of the fatty acid. Monounsaturated fatty acids means that there's a single one double bond, okay? So this structure we're looking at is a monounsaturated fatty acid. Now, the cool thing about these double bonds is that every time there's a double bond in the hydrocarbon part, the molecule bends, okay? So that's how the structure of the fatty acid changes, because every time a double bond is introduced, the molecule bends at that part. So we see that monounsaturated fatty acids have one double bond, therefore they have one bend in that part of the fatty acid. So here's an example. This is oleic acid, which is a monounsaturated fatty acid. So we see this part over here, that's the carboxyl group. And then we have in gray is all of the carbons. The hydrogens are here in white. And just looking at this molecule, we see that, oh, right here, it bends. And so we know that these two carbon molecules right here in gray, there's a double bond that's being shared between those two carbon molecules, and that introduces a bend into the molecule. Now here is another fatty acid structure, and what do you notice here? There are two double bonds present. And so we call this a polyunsaturated fatty acid because it has more than one double bond. Okay, so a single double bond in the hydrocarbon part of the fatty acid, we call it a monounsaturated fatty acid. But if it has more than one, then we call it a polyunsaturated fatty acid. But the same rules apply, that every time there's a double bond in that part of the fatty acid, there's going to be a bend in the molecule. And so here we have linoleic acid, which is a polyunsaturated fatty acid. Again, here's that carboxyl part, and then the rest is a hydrocarbon. And in this case, it's made up of hydrogens in white and carbons in gray. So here's all the carbons and hydrogens, single bonding. Oh, it bends now, right? Here, because there's a double bond. And it continues along. Oh, another bend here as it points down. So there are two double bonds here in linoleic acid. And every time we find a double bond, the molecule structure is going to bend at that point. So just by looking at a structure like this, you can tell whether it's saturated, unsaturated, and if it's unsaturated, if it's monounsaturated or polyunsaturated. Okay, so here's a practice question for you. Pause the video and see if you can insert the correct terms into these three blanks, okay? Your options are going to be unsaturated, monounsaturated, or polyunsaturated, okay? So pause the video, look at the structures, and see if you can put the correct term in the correct slot. Okay, so let's look here. First, myristic acid. If we look at the structure of this fatty acid, we see that it's pretty much linear, right? And so we know if it's linear, there are no bends, that means it's all single bonds, and it is a saturated fatty acid. Oleic acid, we see that hydrocarbon part, right, comes up linearly, but then it bends and turns, but there's one bend only. So it's a monounsaturated fatty acid because it has one turn, which means one double bond. And linoleic acid, as we talked about, because it turns multiple times in its structure, that means it has multiple double bonds, which makes it a polyunsaturated fatty acid. Let's try another practice question. Okay, so I've given you three groups of fatty acids. Now, identify which group are saturated fatty acids, which group are monounsaturated fatty acids, and which group are polyunsaturated fatty acids. Pause the video, think of your answers, and then come back and we'll talk about it. Okay, again, so the key here is that saturated fatty acids, because they have no double bonds, are going to be linear in structure. So if we look, this group are the saturated fatty acids. They're completely linear. Okay, well, how about monounsaturated fatty acids? We know that monounsaturated fatty acids are going to have one double bond. So they're going to have one bend in the molecule. And that would be this group over here. They bend once in the molecule. Now that leaves this group, which are the polyunsaturated fatty acids that have more than one double bond. And so will have more than one bend in their structure. And so we see there's a great variety of structures that we can have here between saturated, monounsaturated, and polyunsaturated fatty acids. Okay, this is the last practice question. Now, instead of looking at these space-filling models, we're looking at line drawings. Now, we see here in this structure, this part over here is our carboxyl group. So that's the part that's going to interact with the glycerol. The rest is the hydrocarbon. So every time you see one line, that means there's a single bond and there's a carbon at each intersection. They just don't draw them all out, okay? So carbon, 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 right? At each point, there's a carbon. And they don't draw out the hydrogens either, but we know, well, if there's a single bond between this carbon and this carbon, there are going to be other hydrogens there as well, sharing their electrons with those carbons, okay? So each time we see a single line, that means it's a single bond between the carbons. Each time we see a double line, that means there's a double bond between the carbons that are there. And then there's more carbon, 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 right? A carbon at every point. So this is the structure, right? The line drawing of a fatty acid. So here's my question for you. This drawing of this fatty acid, is it saturated, monounsaturated, or polyunsaturated? Okay, so disregard the carboxyl part, right? Because we're looking at the hydrocarbon part, the rest of the molecule. And there we see one double bond. So if there's a double bond, we know that it's unsaturated. Now we just have to figure out, is it monounsaturated or polyunsaturated? Well, it has one double bond, so that means it's monounsaturated. Try this one next. Okay, so here's another structure. Is it saturated, monounsaturated, or polyunsaturated? Now, again, you're going to disregard the carboxyl group on the end. That's the part that's going to bond to the glycerol. And we're looking specifically for our rules here at the hydrocarbon part, the rest of the molecule that's only made up of carbons and hydrogens. And here, maybe you said polyunsaturated, and that would be correct because we have 2 double bonds found in this part of the molecule. 2 double bonds, that means polyunsaturated because polyunsaturated means more than one double bond. Okay, last question. How about this one? Well, this is a saturated fatty acid. We have the carboxyl group over here, but then we see the rest of the molecule, there's just single bonds between each of those carbons. And because there's single bonds and no double bonds, we call that a saturated fatty acid. So remember, bringing it back, we've been talking about the fatty acids. Triglycerides are these molecules that are formed when this glycerol molecule bonds to 3 fatty acids, okay? And that structure is known as a triglyceride. That's the main structure that we find neutral lipids like oils and fats in. Let's look at this triglyceride structure for a minute, because we see the 3 fatty acids in it are all different, and that frequently happens. But let me ask you, are the fatty acids attached in this triglyceride, are they saturated or are they unsaturated? Right, they're unsaturated because we see that there are bends in each one of the fatty acid molecules attached. So this is a triglyceride that has 3 unsaturated fatty acids. Now, the cool thing about these bends or these double bonds is the more double bonds in a molecule, the more fluid it is. So this triglyceride has 3 linear fatty acids attached within the molecule. Now, if we look at the other triglyceride shown here, we see bound to that glycerol, we actually have benzy fatty acids. So in this particular triglyceride, we have 1 saturated fatty acid, 1 monounsaturated fatty acid, and 1 polyunsaturated fatty acid. And the more bends in those fatty acid tails, the more liquid or fluid that molecule is going to be. The reason for that is here, because if we have 3 saturated fatty acids in a triglyceride, they can pack together really tightly, right? All these triglycerides can pack together really tightly, forming a more solid substance. But if there are more double bonds and therefore more bends in the fatty acids of the triglyceride, that means that those triglyceride molecules, when they're together, are going to space out farther from each other because those bends create more space in the molecule. So the more bends that there are in those fatty acids, the more fluid or liquid the substance will be. So oils are liquid at cell temperature. Fats are semi-solid at cell temperature. So what does that tell you about oils versus fats? It tells you if oils are liquid, they must have more unsaturated fatty acids in them. And fats, being semi-solid, they have more saturated fatty acids within them. Okay, so the reason that oils are more liquid is because they have more unsaturated fatty acids in their triglyceride structures, leads to a more fluid substance. On the other hand, fats have more saturated fatty acids, which allows them to pack in tighter, creating a more solid structure. So the more unsaturated fatty acids that there are in a particular substance containing triglycerides, the more fluid that substance is going to be. Here's an example for you. Butter, which is a fat, has 54% saturated fatty acids. And that's why it's solid, right? It's semi-solid. It has more saturated fatty acids in its triglycerides. Olive oil, on the other hand, has very few saturated fatty acids in it. In fact, 81% of the fatty acids found in olive oil are unsaturated. And it's kind of interesting that 70% of the fatty acids found in olive oil are actually monounsaturated. Okay, so oils are going to have more unsaturated fatty acids in the triglyceride structures. Fats, like butter or lard, are going to have more saturated fatty acids in them. So this is a summary again of what we talked about today, right? Fats and oils are neutral lipids. Fats and oils have a structure known as a triglyceride. Triglycerides are made of that glycerol backbone that ends up being bonded to 3 fatty acids. And those fatty acids can be saturated or unsaturated. If they have no double bonds in the hydrocarbon part of that fatty acid, it's known as an unsaturated fatty acid. If they do have 1 single double bond, well, that makes it a monounsaturated fatty acid. And more than 1 double bond in the fatty acid makes it a polyunsaturated fatty acid. So just for fun, the next time you're looking at food and you've picked up a box and you're looking at the nutrition label on the side, you can actually look to see how many grams of saturated or unsaturated fatty acids are found in that food. And some labels will be nice and break down the unsaturated into monounsaturated and polyunsaturated. Because remember, these biological molecules, like fats and oils, are molecules that make up living things. That means us, but that also means the plants or animals that we end up eating as food as well.